Copyright Stanford University, all rights reserved.
Okay, we're going to get started. Welcome, everybody. Glad you could make it today. Joe's out of town, so it's my pleasure to give the announcements this morning. On Wednesday, the Med 223 is started up again for the year 2 or 3 sessions ago. We'll have Dr. Joseph Wu, the chair of cardiothoracic surgery, going to be speaking on cardiomyocyte regeneration at 12 noon tomorrow at the Falk Library. Next week for Frontiers, we'll have Dr. Dahlman, the chief of vascular surgery. He'll be speaking on abdominal aortic aneurysm disease, pathogenesis and progression. Now, Frontiers is actually now being recorded, depending on the permission of the speaker. Sometimes we do not do the recording if there's, they rather have it not released to the wide world. You're welcome to check out the CVI's YouTube page at tinyurl.com/CVI Frontiers. We only have one talk up there right now, which is Dr. Vagilos's, which was the largest turnout we've ever had in this room, over 180 people. It was very well attended. If you were in the far back and like to actually see what it was like inside, visit the site. Yesterday we had our first clinical biomarker discovery training program hosted by Dr. Rajadas. The next one is going to be the first Monday of every month. Is going to be 5 p.m. November 3rd. And to register, you can visit this website. You can also register now for the 2014 Cardiovascular Institute Annual Retreat, which will be December 2nd, all day, pretty much from 8:30 until 5:30. The keynote speaker will be Douglas Mann, the Chief of Cardiovascular Division of Washington University. The CVI is doing its travel awards, so please submit to Ingrid when you get a chance by December 15th. So you can travel, exchange your ideas, and help you go to conferences. This is particularly geared for postdocs. The manuscript award, what we do yearly, is due on January 15th. So if you had a manuscript that's been published this year, the year of 2014, in a journal, submit it. Again, Ingrid is running this, and the top 3 will be giving awards. And this Saturday, in connection with Nick Leeper, and the AHA, we're hosting. Nick Leeper's group is going to be hosting the 2014 Silicon Valley Heart and Stroke Walk. We have a booth there. The Stanford Hospital has a booth, and the Cardiovascular Institute has its own team. And I met my own personal fundraising goal thanks to some people in this room. I appreciate that very much. So come out and join us at 8:30. And now I'm going to pass it to Ingrid to introduce our guest speaker today.
So today we have a treat. Our speaker received a degree in biochemistry here at Stanford. He completed his fellowship at Duke, and now he is the vice chair of clinical research at the School of Medicine. Ken Mahaffey.
Thanks. Appreciate it. Thank you very much, David. Well, good afternoon, everyone. It's really a pleasure to be able to come and spend some time with you and talk about something that I do every day, and that's clinical research, but more importantly, trying to figure out how to help other people do their clinical research here on campus and actually nationally and internationally as well. Just like to acknowledge my current financial disclosures. For the last 10 years, all of my financial disclosures have been on the World Wide Web, both at the Duke website shown when I was at the Duke Clinical Research Institute, and now here at Stanford at the website shown. Well, I want to start off today providing a foundation or a groundwork so that we can all be on the same page. And that really is why we do clinical research. And since this seminal depiction by Caliph and colleagues many years ago, There's been others that have described similar types of cycles of clinical therapeutics. But I like this one because it really integrates a lot of the things that everyone in this room does. We're all here to think about novel interventions or novel concepts, and this could be basic discovery of a new molecular entity or a new biomarker or a new drug or a new device, or even maybe a new way of delivering healthcare. Once we have that concept or idea, we then need to study it in clinical trials. And I would suggest that, at least up to now, we've done an okay job of that. We have a long-standing tradition of being able to develop clinical trial infrastructure to do these 10 and 20,000 patient clinical trials. But they're all individual silos of infrastructure and effort. And then after each trial is done, we dismantle them. And so we need to think of ways, and I'm going to spend a part of my talk introducing you to some novel ideas and methods to do that kind of research. We take the data from the clinical trials, data that show that certain interventions are safe and effective. We then try and incorporate in our practice guidelines. That's what, in our field, in cardiovascular disease, what... Hello? In cardiovascular disease, what the ACC, AHA, and other subgroups of those important scientific congresses tell us that we should be doing. Patients who should be treated with aspirin should get an aspirin after an MI. And we now have a better strategy and I think are more efficient in terms of updating those guidelines for clinical practitioners. But now in the last few years, we've now developed ways to track performance. Are physicians and clinicians actually doing what they're supposed to do? And now, as you know, if we're not, CMS won't pay us. And so we hope that we can improve outcomes. And now there are a multitude of investigative groups that have shown when you do what you're supposed to do, outcomes improve. And that really completed this cycle of clinical therapeutics. Now we just need to continue the cycle to continue to improve care. I would argue again, and it's easy to make this statement in a cardiovascular institute meeting, but when I'm lecturing in other about other therapeutic areas, I get a little bit nervous. But I would argue that cardiovascular disease is further along than any other therapeutic area in medicine in generating the evidence, the empiric data about what we're supposed to do to treat patients. This was a really interesting paper from Tricoci and colleagues published now 4 or 5 years ago in JAMA. And what it shows is the level of evidence associated with recommendations in the guidelines. These are all what we call Class 1 recommendations. In our guidelines, there's Class 1, Class 2, and Class 3. Class 1, you're supposed to do it because it's beneficial. Class 3, you're not supposed to do it because it could actually hurt patients. Within the classes of recommendation, there's level of evidence A, B, and C. Level of evidence A is evidence from randomized clinical trials or very large registries in conjunction with smaller randomized trials. Evidence B is a little bit below that, and C is just expert opinion, right? Someone with maybe a little less hair than me, a little more gray hair, or perhaps the classic definition of an expert, right, is a guy from out of town with slides. But look at these data. Only 20% on average. 20% of the things we're supposed to do have level of evidence A. Right? That means that 8 out of the 10 things we do for our patients across the street in the CCU, we're doing it because we just think it's the right thing to do. There's no evidence, definitive quantitative evidence to support what we're doing. It's pretty humbling. I play that game with the fellows when I'm in the CCU or on rounds. Is tell me what you did for this patient based on the evidence from the guidelines that is Class 1 level of evidence A versus everything else. And the list is always longer. We just do what we think we should be doing or what we've been told over the years. So we need to come up with better strategies to develop this evidence. And why? It's because even though we want to do the best thing for our patients, often our intuition is wrong. And I think many of you have probably seen slides like this, or in fact, maybe this slide in other lectures that I've given. And this is a very brief list. You could all come up with other things likely in your area of cardiology. But this shows well-conducted randomized trials that were performed with treatments that were thought to be beneficial in a variety of different disease states. But when the large randomized trials were completed, these drugs actually hurt people. And as in the majority of them, they were shown to increase mortality in these patient populations. So even though I ran around as an intern and a resident in the early 90s trying to stop all the PVCs on the monitor in patients with myocardial infarction, the nurses were happy, I was happy, and we thought the patients were happy when randomized trials showing that decreasing the premature ventricular contractions in the CAS trial was actually done. It showed that patients who got those drugs did worse. We don't do that anymore. But we wouldn't have known that unless these trials were done. But what's the challenge? Well, the challenge is that we often have to rely not on clinical outcomes, but we have to rely on what we call surrogate markers. Surrogate marker in the example I gave was a PVC, an extra heartbeat. We thought a lot of those extra heartbeats was a bad thing. Well, they might be a bad thing, but the problem is the intervention that we use to treat them was not a good thing. And this is, if any of you are interested in thinking about surrogate markers and biomarkers in clinical research and clinical outcome study, this is a phenomenal paper to pull out from many, many years ago. I'm not going to tell you to read many things today, but this is one of them. And this here shows when a surrogate endpoint or a biomarker actually has some validity. And that is when the pathogenesis of a disease process and its relationship to the clinical outcome is all contained within that surrogate outcome, and the intervention that is being studied has all of its effects, both good and bad, on that disease pathway. I challenge you. Can you name any one intervention or disease process that would look like this? Because this is what typically happens. The intervention may have absolutely no relationship or effect on the surrogate endpoint that we're studying. Or the intervention may have multiple targets, both on the surrogate endpoint that we're measuring in the clinical study, but also may have other effects outside or involved in other pathways of the progression of disease and its relationship to the clinical outcome. So it's complicated, and therefore relying on individual surrogate markers or biomarkers to make decisions about clinical care is not a good thing. I would argue, though, that it's a very good thing to do when we're trying to identify novel therapies and interventions in early phases of development, because we can't do big trials. So we need to rely on thoughtfully collected surrogate endpoints or biomarkers to try and identify promising therapies and interventions. But we always need the large outcome trial to be confident in the balance of the good effects and the bad effects. Because with any intervention, there's always a balance. And it's what happens when you mix the balance of the good stuff and the bad stuff and how it relates to final health outcomes in our patients. So I'm a clinical trialist, so I'm a little bit biased. But I think that the conduct of large randomized trials or large outcome studies is what we need to continue to do in the future. Do we need to do them differently? Absolutely. Because right now, the way we're designing and running these large clinical outcome trials and registries and studies, it's increasingly complex, it's increasingly costly. We have this whole concept of good clinical practice guidelines, but I often argue that they're not good guidelines, they're not clinically relevant, and they're typically not even practical. We need to do away with old policies, guidelines, and procedures and think of better ways to still protect human subjects who are volunteering to participate in human experimentation, but we need to think about how to do it in this century and not prior eras. And obviously layers of ethic approvals where we have IRBs all across the country reviewing the same protocols needs to be blown up and rethought. And I like this quote from this article. And basically it sums up what is clear is that such processes are extremely expensive and delay studies. So again, I think we need a revolution of innovation in how we conduct these large clinical trials. But this is not a
2 years before she realized I just restarted.
Where are you taping this? Or where are you sending it?
Live stream to Hawaii.
Great.
It looks like they turned off.
No problem. That's terrific. And as you'll see in a couple comments later on in the talk, I am fully supportive of having it videotaped and displayed everywhere in the world. So this concept of trying to do things differently is not new, but I would suggest that it's relatively new in our thinking process in the area of healthcare research and healthcare delivery. This is Edward Deming. He was actually a statistician and an inventor, and he helped revolutionize the automotive industry. Unfortunately, even though he was an American, no one wanted to listen to him in the United States in the mid-1900s, so he went to Japan and helped them revolutionize their automotive industry. And he had this whole concept of relying on the evidence that you collect from the operations and the metrics of how you process things. And I like to apply this evidence-based operations to how we do clinical research. That is, measure what we're doing. And is it having an impact on doing the research? Is it improving efficiency? Is it improving safety of the human subjects? Is it improving the quality? Is it decreasing cost? And actually analyzing what we're doing in a quantitative way, the way we all analyze data from animal experiments and clinical experiments all the time. But let's apply it to the healthcare domain and clinical research. And I think there's increasing pressure that we need to do this and are starting to do it on a broad scale. And that's because, as everyone in this room is acutely aware, the funding sources for doing research is quickly dwindling. We had Michael Lauer here from the NHLBI several months ago who described very nicely the challenges of funding mechanisms through the NIH and other government agencies at the federal level. Here you can see the data from biopharma and research and development. And remember that of about the $200 billion that are spent every year on clinical research and R&D in the world, that about 75% of it comes from industry. And it's fortunate, at least for the projections for the next several years, that there will be perhaps a modest but not very large growth in R&D. Unfortunately, I was one of those people that was stuck way out in the hallway last week and couldn't get in the room. So I don't know if Dr. Vagelos had a different perspective on things, but I haven't had a chance to download his talk yet. What about here in terms of extramural support for research at Stanford? Harry Greenberg, Senior Associate Dean for Clinical Research in the School of Medicine, and Chris Webb, who runs the CTSA or Spectrum, provided these data for me that really show the heterogeneous funding sources that we have here at Stanford. But you notice that it's in the bottom left quadrant here that only about 20% of the research funding on campus is not from federally funded or government sources. And so I think that as the dean is continuing to promote the need for more clinical research on campus as part of his priority, we're likely going to have to invest in alternative strategies for developing funding for clinical research and other types of research on campus, including stronger perhaps ties with industry, non-profits. Obviously here philanthropy is a great source as well in the Bay Area. So I think we need to rethink some of our strategies. What about specifically in the School of Medicine, where many of us have our homes? Here are data that Tom McGrath, the financial person from the Department of Medicine, shared with me a few months ago. And within the School of Medicine right now, there's about $500 million in research expenditures. And you can see that only about 4% of those expenditures are on clinical trials. That is enrolling Stanford subjects or patients in research here on campus. In the Department of Medicine, there's about $100 million in research expenditures. And about 10% of it is on clinical trials. That's new interventions, drugs, devices, and services for Stanford subjects. And so I think we have a lot of opportunity for growth both in the department and the school for research activities. This was an interesting article when it came out a couple years ago. And there's been, great discussion and debate about the role of academics in industry, and particularly in industry partnerships for performing clinical research. And I think that obviously there's a need to have these partnerships, and it's a little bit of a unique entity here in the United States. As you can see from this graphic, there is a substantial number of relationships with academia and industry that have led to productive ventures in terms of drug development. But as you can see, looking across a number of other countries, it's relatively unique. And I still think these partnerships are important, but I think they clearly need to be governed by appropriate guidance and policies to protect us both with institutional conflicts of interest, but also individual and personal conflicts of interest, so that we understand the transparency of these relationships, but also the importance of what can come from them in drug development and discovery. The next few slides, Mike Lauer was nice enough to share with me from his talk in Medicine Grand Rounds earlier this year. And I think they summarize so nicely I couldn't even improve upon them. And I asked him if I can show them just as he had put them together. And this is really the classic clinical trial business model that I described for you a little bit earlier. And that is, we develop small individual infrastructures and silos to do each of these trials. And because of the cost of that, when you look at clinicaltrials.gov, in an analysis that was done by Caliph and a number of other people, including Deb Zarin, who, as you know, spent, I'm not sure if she's still here or not, but she spent sabbatical time here in HRP thinking about some of these issues. And when they did an analysis of the types of trials that were registered on clinicaltrials.gov, they found that most of them were really small. We're talking hundreds of patients. And it is really hard to make real quantitative empiric observations and inferences, particularly about interventions, when you're talking about sample size that small. And most of these, because they had to develop all their own infrastructure to perform the research, even though they had small numbers, they still were quite costly and had high budgets. We've already talked about the challenges and limitations of surrogate markers. And basically what his conclusion was that this research enterprise is really a parallel universe to our clinical enterprise because of the independent silos that are created. something else to pick up to read. So this is only the second homework assignment I've given you. This is a terrific book to read, The Innovator's Dilemma. And it really talks about disruptive technology and how we need to embrace it. And even though it may not feel comfortable when we're trying to challenge the norm, in the end, it can actually be incredibly beneficial. And Mike's takeaway from taking those philosophies, and I agree with him, is that what we need to do now is try and leverage the infrastructure that we've created to do clinical research and ask more questions on the backbone of the research infrastructure that's already in place. And this can generate an incredible amount of excitement. I think that when Bob Harrington was here previously, he described for you that in one of the large acute coronary syndrome trials that we created, we built in a database repository for DNA, RNA, plasma, and serum samples for biomarker assessment. And now we have this incredibly rich repository that was very easy to collect because it was done on the backbone of a large randomized industry-sponsored clinical trial. And I'm working with several of you here to actually help a simple trialist like me. What do I do with all these samples? And so now we don't have the challenge of collecting them. We have the challenge of what do we do with them? But it was the incremental cost to collect 10,000 patients worth of blood samples and already have them well clinically phenotyped within the construct of a clinical trial and have over 1 year of follow-up on them for endpoints. It's an incredible database that we're looking forward to working with many of you on. So what we need to do in the new era, as Mike nicely described, is that I think we need both bigger trials that are simpler and easier to do, but embed within them smaller mechanistic studies that can help us understand both the underpinning of the observations about the interventions that are being studied, but more importantly, about the diseases that we're studying. And I think that we have great opportunity now as we move forward in the electronic era, the digital era of data resources, to be able to partner not only with other academic groups, but now with healthcare systems that have electronic health records, and even the patients that are getting care to engage them in the research process. Well, what about the research process here at Stanford? Well, just this week, we're launching the Stanford Center for Clinical Research, or SCCR. It's a center that is going to sit within the Department of Medicine. And we're going to try and provide researchers here who want to do clinical-facing research with the infrastructure to help them do that kind of research. And within the Stanford Center for Clinical Research, we're going to have 3 major enterprises that you're going to be hearing more about. The first is site-based research. And that is enrolling Stanford subjects, Stanford patients in clinical trials, whether they're observational registries, whether they're interventions or health services research that's engaging at the patient level. And then a data coordinating center enterprise that will focus on helping Stanford faculty and researchers who have an idea and want to develop a protocol and then enroll patients, need help with setting up data platforms, help with statistical support, perhaps some core laboratory support, and actually not only help design the research, but help you do the research. And then finally, a third enterprise of education and training. And as we get more and more people engaged in clinical research, we need to make sure that they have the tools in order to do good quality research and protect the safety of human subjects. Our mission, therefore, is to support the faculty, but also help the faculty and promote and innovate high-impact global clinical research. And notice I don't say clinical trials. It's broadly defined research in the clinical arena with the ultimate goal of improving the patients that we see every day. This is the leadership team that is growing quickly. We're going to be partnering with Manisha Desai, who many of you, I think, know in the Quantitative Sciences Unit, which is part of the Department of Medicine in the division of BEMER, or Medical Informatics. And I'm really pleased with the partnership that we're going to have with her and with Mark Musin. We have an associate director, Rebecca McHugh, who is going to be leading the site-based research enterprise. In fact, Rebecca's in the back of the room. So if you have a need or want to understand more about what we're doing, I'm sure she'd be delighted to talk to you. And Amal Jamani, actually his second day, is in the back of the room as well. I just saw him, sorry. Has just joined us from Texas, where he spent many, many years growing a research enterprise there in the Texas Heart Institute. And finally, Rhonda Larson, who is spending time with us in the Department of Medicine. She's a world's expert in setting up site-based research enterprises and is helping both Jam Gahar in the Brain Trauma Center, but also us here in the Department of Medicine. This is the organizational structure, just to put it in perspective, with the site-based research enterprise under Rebecca's leadership, the coordinating center enterprise under Amal's leadership, and the partnership that we're going to have with the Quantitative Sciences Unit. And I'm not going to go into great detail about the kind of services and research we want to be doing, and perhaps I'd be happy to chat with people either after the session today or separately later on. We have some key fundamental missions. That is, we want to have high impact in the academic environment. We want to be doing important research that's generating important knowledge about disease, about the delivery of healthcare, about new drugs and devices, about population health sciences. We want to invest in our own people. We want to invest in house staff. We want to invest in fellows, postdocs, and young faculty in particular, to help train them, to help them do good research so that they can help promote their own careers. I've already talked a lot about operational effectiveness, evidence-based operations. We want to adhere to those fundamental principles as we design procedures and strategies to do the research. And then obviously in any academic environment, being financially accountable for what we do is important. Well, how does this fit? How does the Stanford Center for Clinical Research fit within all of the other resources that many of us have available to us on campus? From Spectrum under the CTSA to a number of important institutes like the Cardiovascular Institute, to other departments that are doing this research. We at the Stanford Center for Clinical Research do not want to compete with these important entities and resources that you have, but rather complement them. And I think many of the resources here can help people design and navigate through the Stanford system. But what the Coordinating Center can help do is actually do the research, provide the resources, provide the information technology to do the research that you want to do. When we look across the Department of Medicine, we did a survey to see how many faculty were actually involved and how many coordinators were involved in clinical research. And you can see here that across the department or the divisions within the Department of Medicine, there are a number of faculty and coordinators involved. And in fact, about a third of the department faculty conduct some sort of clinical research. So we think there's a growing need for these kind of activities and support. Here's the number of IRB protocols across the number of divisions. And I hope as I continue to have partnerships with Joe Wu, Francois Haddad, Sean Wu, and many of you in the room, that we can actually grow the cardiovascular portfolio here, both in the Cardiovascular Institute as well as in the cardiovascular division. But through this exercise over the last 4 to 6 months, we've also recognized a number of barriers for doing research that we think in the Stanford Center Clinical Research Center that we're going to be poised to help break down. And that's helping people have platforms to track the finances. This isn't just necessarily the cost of doing the research, although that's part of it. It's how do they track the finances? How do we track patients in clinical trials versus patients in clinical care? Providing more coordinator support, and I know Joe Wu here is already working on hiring people to help provide coordinator support for researchers here in the Cardiovascular Institute and across campus, and a number of other things in grants and contracts that we've heard from before. What kind of research are we already doing in the Center for Clinical Research? Well, here's an example of some of the awarded grants that we've had over the last several months. A number of projects with Manisha Desai looking at databases. So many of the house staff and fellows are already coming to look at opportunities to do analyses on already established databases, to write papers, which is obviously very important as they look for fellowships in academic environments or faculty positions. We have an important program with Bill Fearon with St. Jude and Medtronic. It's actually an investigator-initiated program to look at fractional flow reserve, measuring the flow through coronary arteries. Tomorrow at Grand Rounds at the Department of Medicine, Jam Gahar, a neurosurgeon, is going to talk about his brain trauma research, so come and you'll hear more about that. Paul Wong in EP has a large ECG core lab, actually the largest ECG database on hemodialysis patients, over 4,000 of them. It's going to be a rich repository for a number of analyses. And just about 10 days ago, PJ Utz in rheumatology got a nationwide network grant that we helped put together in the Center for Clinical Research to coordinate rheumatoid arthritis and lupus research across the country in 15 sites. And I think there's actually going to be great opportunity here because much of this is targeted at basic discovery with tissue samples in these patients with inflammatory diseases as well as blood samples. And we all know the intense research that's going on in the overlap of inflammation, anticoagulation, and cardiovascular disease. And I think a great opportunity for us to bring a number of different research groups across campus to think about what we want to investigate in this important network. We also have a number of projects that have also been submitted. And again, the theme here is that we're partnering with a number of different faculty members across a number of different therapeutic areas, as well as a number of different funding sources. So a very heterogeneous profile of research. And I put this up there to show you what we're doing in the center, but also for you to get an idea of what kind of research is being done. So if you see an investigator that you've worked with before or a therapeutic area that you may have interest in, these are the people to come and talk to, where we can leverage relationships to do important research on the backbone of what's already being done. Just to highlight, this is PJ Utz's network grant that is really looking at 15 centers across the United States that is going to collect clinical data, tissue data, and blood data on these patients with inflammatory disease and really develop a sustainable research enterprise to continue this kind of research in the future. I'm going to skip on this because I have a feeling Dr. Gahar is going to show this and talk about it more tomorrow morning with his Brain Trauma Foundation. One research project that many of you may have heard about is the Google Baseline Project. This is a partnership between Duke University, Stanford University, as well as Google. There were a series of press releases that came out over the summer from the three institutions. This is a program that's being led by Sam Gambier, Chair of Radiology. The operations of this effort that I'll describe in just a second are going to be run through the Center for Clinical Research under Rebecca's leadership, and we're excited about that opportunity. We're working with Dave Marin, who is one of the new faculty in cardiology, to lead the cardiovascular effort. George Sledge in oncology is going to be leading that effort, and we're working with Mike Snyder for genetics expertise. What is this project going to do? This project is going to look at enrolling 10,000 patients between Duke and Stanford 5,000 cancer patients, 5,000 cardiac patients. A third of them will be normals, a third at risk for disease based on traditional risk factor assessment, and a third with known disease. And we are going to do a comprehensive, very rich and very deep phenotyping, genotyping. We're going to collect microbiomes, echocardiograms, x-rays, imaging studies, and then follow these patients longitudinally with the hope that over time we can identify novel strategies to predict patients who may develop disease, but also better ways to predict future path of disease progression. And obviously, Google's interest in this is try and overlay on top of traditional risk factors and assessments of disease that we use now in clinical practice, different technologies to try and identify signals of disease. So this is going to be an incredible repository of information across a large cohort of patients. In fact, the largest cohort of patients that's going to be this deeply phenotyped that's ever been done. And we'll have great opportunities for research. And we've engaged with many people in the Cardiovascular Institute about helping us think through what we should be collecting now and what we should be storing for later analyses. So I look forward to hearing more ideas and engaging with more of you in the future. Just want to highlight that as part of the Stanford Center for Clinical Research, part of our mission is education. We're going to be having an educational session in November in conjunction with the Cancer Institute. We're bringing in outside speaker Lisa Zimmerman, who has a national reputation for being a fantastic educator, teacher, and speaker. And it's going to be a program to help people understand what it means to be a principal investigator or a researcher when you're engaging with human subjects. So if you have an interest in this area, please sign up and come and join us. I want to just shift gears for about 5 more minutes here, and then we'll have time for questions at the end. It's fun to think about all this new Google stuff and collecting samples to understand disease better. But I think that, as Bill Gates said several years ago, some of the biggest advances we're going to make in the next 5 or 10 years are actually going to be taking the things we already know and figure out how to apply them or engage those discoveries in how we care for patients. A couple of simple examples. Many of you may have seen this in Jack recently, that we know there are important medications people need to take after they have a heart attack. In fact, there's anywhere from 5 to 7, depending on how big their heart attack was. We also know that the more medicines or more pills that people need to take, the less compliant they are. And the less compliant they are, the worse their outcomes are. So what a simple idea. Just put them all into one pill because everybody's taking the same 5 medicines. This was actually a poly pill with 3 different medicines. Now, it sounds easy just to pack the pills all together, but you got to be careful about maybe interactions. You have to be careful about absorption when you put the pills all together. So they did clinical trials that showed improved adherence when you did a simple thing like putting the pills all together. Pretty simple idea, now shown to help in patient outcomes. What about how we do research? This is an example of a trial that was done primarily in Sweden called the TASTE trial. Why it was done and the results is not why people are going to remember this trial. It was how they did it. The trial was looking at whether or not if you sucked out clot from the heart artery in the middle of a heart attack, could you improve outcomes? It showed there was really no difference in 30-day mortality. But the way they did this study was not a standalone database. They have a registry that captures every patient that goes to the cath lab in Sweden, in every cath lab. And all they did was add 1-page module on top of all the data they already collected. And they did the study on the backbone of that database that was already in place. Why was that so important? Well, for a couple reasons. You can see here they enrolled 7,200 patients out of 11,000. when we do clinical trials in a standalone database, we maybe get 1 in 10, 1 in 20 at the most. So talk about the ability to generalize the results to the broader population. Number 2, by using the registry, no patients were lost to follow-up because they had all their clinical care followed routinely as part of the registry, an electronic health record. And then number 3, the cost. Usually when we do these trials as a standalone trial, this could be $10,000 a patient. This was a couple $100. So very efficient, easy, and cheap. And we need to be thinking about how we're going to do more research like that. Here, and we have experts on campus that are already thinking about these things and already publishing on these things. Here's an article from Mei Zhang and Philip Labori about looking at important methods that we can apply to do point-of-care randomization. And I'm going to skip through this just to keep things moving here. You've heard a lot, you've read a lot, both in the lay press and in the scientific literature about learning health systems. And we need to make this a reality. And with electronic health records, with information technology, with methodologists that can help us think through the importance of randomization at the point of care, I think we're going to get to the learning health system as described here. And we're already doing this. With the electronic health record that we have across the street, with Epic, you can see here that investigators have actually published on how they made important patient decisions, leveraging the data that was available at a given time. This was an example from Chris Longhurst and colleagues about whether or not to anticoagulate a young patient that came in with immunological diseases and an inflammatory process that was leading to clotting. And no one knew if anticoagulation was the right thing. And they looked back at the prior clinical care at Stanford Health Systems and at Lucille Packard, and then published here in the New England Journal. I mentioned early on I was going to talk about this. I got these slides from Mike Gibson because I think they're absolutely spot on. And I said earlier that I had absolutely no problem sharing my slides or having this videotaped, and I think that's important. Because when we're doing important research and we're talking and learning about important topics around clinical research, we should be sharing this knowledge. And historically, it's been copyright, it's been patent. We got to protect all our discoveries. We need to be in an environment and a culture that embraces that. And that's sometimes challenging in the academic environment when our promotions and our grants may be dependent on having that knowledge protected. But I think as we move into the new world of learning health systems, we're going to have to find ways to overcome that and recognize team science. And as Mike often says, it's copyleft, not copyright. So I'm going to conclude here. As you're thinking about all the kinds of research that you do, whether it's basic discovery or it's phase 1, phase 2, or phase 3 research, and we clearly think about this in the Stanford Center for Clinical Research, think about the importance of the research that you're doing. Challenge the science, challenge the methods, challenge how we're doing the research every day. Because if we're doing good research, we're going to be making an impact. And if we're making impacts, we're going to be improving the care of patients around the world. So thank you very much for your time and attention today. Appreciate it. Yeah, so I think I can answer it 2 ways. One is, are there any biomarkers that actually...
This meeting has ended due to lack of activity.
Oh, that hurts. That hurts. Yeah, so part of it is, are there really biomarkers that have been validated? And I think probably the closest one that we have in clinical cardiology is LDL. at least for all the drugs that have been studied so far, we see clearly that that is an important measure for outcomes and also benefit, but not all the benefit, because we still think there may be pleiotropic effects for statins. I think we're really going to see the test of that biomarker as the new class of superstatins. They're called PCSK9 inhibitors that actually drive LDL down to what I call caveman levels. We're driving LDLs down to the 20s. And whether or not the LDL hypothesis will hold up when you're driving it down that low is unclear. You know, in an area that I spent a fair amount of my time for a number of years, I was looking at reperfusion injury. That is, when you open up a heart artery in the setting of a heart attack, there's very good things that happen, but there's also deleterious things happen. And that field has had a number of clinical trials that have used biomarkers of infarct size, of biomarkers of inflammation and coagulation, that we looked at one or two of those markers and then just found that they improved. And then we did big studies, and we never found a drug that worked. But I think that the strategy moving forward, and this is being implemented more and more, is you measure multiple biomarkers. And you look at multiple biomarkers that you think might have a relevance or association with the biology of the disease and the intervention that you want to study. And you don't just rely on one or two. You have a series of them, and then you figure out how many are going up and down. And if there's more thumbs up than thumbs down, maybe you move ahead. Well, I'll let people go, and I'm happy to stick around and hear. Mark, yeah. So I think you're exactly right. It's being tackled on a variety of levels here. At one level, we're actually trying to do that research. As you may know, within EPIC, there is actually, you can embed modules to identify specific patients. It all started with a researcher who wanted to do a clinical study on dog bites. And so every time dog bite diagnosis went into EPIC in the emergency department, a pager went off and they could run down and see that patient. And now we have a number of tools. But that's a very simple example. I think that there's three areas that we need to engage in. One is we just need to make that the culture. And I think we're starting to get traction both at the School of Medicine and at Stanford Healthcare at the leadership level, that is an important priority. Number two, we need the information technology to be able to engage with the research activities at the point of contact of the physician and the patient. And right now there is a information or informatics task force that's been called by the dean and the leadership of the three hospitals that's being chaired by the CIOs of each of those institutions. And one of the priorities is to figure out the information technology that is needed to do exactly what you described. And then the third area is that we need to engage the patients. And we're doing that on a couple levels. In fact, a woman named Bella Chu in general medicine disciplines has what's called the Stanford Research Registry. She's now gone and done what you suggested, and that is collected about 1,500 patients that have simply signed a consent that they would love us to call them if we have a clinical study that may be appropriate for them. She's actually stalled because she had a lot of difficulty getting the people who were the intake personnel. When you walked in the clinic and they say, hello, nice, glad you're here to see Dr. So-and-so, they didn't really want to give the consent form and just have the patient read it and sign it. So we need to change that culture, but those kind of things are ongoing. And then finally, there's actually a platform called Choir that's been developed. I don't know a whole lot about it, but by Sean Mackey in anesthesiology, that can actually help that be done in a more seamless way. And we're already partnering with him to try and figure out. Rebecca's actually reaching out to both Sean Mackey and Bella Chu to see if there could be some synergies there to help continue to grow that research registry. Great. Okay, thank you, everyone.